Hello. Can you hear me?
Hello. Hello, Kashyapi.
Sir, I can hear you, sir. Good evening.
Good evening. Good evening. Yeah.
Hello.
Hi.
Dr. Ghosh, can you hear me?
Yes, I can hear you. Yeah.
Hello.
Hello. Yes. Can you hear me? Hello.
Hello.
Who's that?
There is some problem at his end.
Kashyapi. Balchandra here, sir.
Kashyapi, can you hear me?
Yeah, I can hear you. Can you hear me?
Yes, I can hear you.
Okay, fine.
We can hear you, everybody.
Good evening.
आता मुलगा येईपर्यंत थांबतो मी कुठेतरी. तू फक्त आयसीयूचा बेड बघून घे. तसं बघा अजूनही त्या सांभाळत काय ना असं सारखं टांगती तलवार ठेवण्यापेक्षा एकदा होऊ दे.
आम्ही शरणागत सरांकडून आता तुमच्याकडे म्हणजे ट्रीटमेंट वगैरे घेतली आता तिला कसं एक्झॅक्टली आम्ही नेलं ना आयसीयू मध्ये नवीन हॉस्पिटल असणार ती थोडी अजून घाबरेल.
नाही होणार. आपल्याला मेडिकल कंडिशन जास्त महत्वाची नाही का? असं काय करते तुम्ही? म्हणजे हे बोलणं अगदीच हे आहे, तसं नाही म्हणून चालणार. आता आपल्याला जर का याच्यामध्ये सिटी स्कॅन मध्ये जर का तिथे कन्सॉलिडेशन दिसतंय तर थोडंसं हायर अँटीबायोटिक आणखीन काय करायचं का हे बघायला तिथे गेलेलं बरं स्टेबिलाईज होईपर्यंत. बाकी तर किती स्टेबल आहेत हे आपण बघतोच आहोत. त्याच्यामध्ये कुठेच काय असं झालेलं नाही की बा कुठे काहीतरी मोठा प्रॉब्लेम झालाय, ब्लिडिंग झालंय किंवा काहीतरी कॉम्प्लिकेशन झालंय, काहीच नाही. हे आता याच्यामध्ये लंग्स मध्ये इन्फेक्शन होणं या वयामध्ये एल्डरली पेशंट मध्ये आता काय अचानक होऊ शकतं थोडस. म्हणून आता बरं दुसरी गोष्ट म्हणजे आपण ऑपरेट केलंय लेफ्ट साईडला, त्यांना कन्सॉलिडेशन म्हणजे जो निमोनिया टाईप झालाय तो राईटला आहे. हा ते लेफ्टला नाहीये.
म्हणजे निमोनिया
राईटला सुद्धा थोडंसं फ्लुइड आहे पण ते रिॲक्शनरी असतं म्हणजे ऑपरेट केल्यावर थोडंसं येतं. मी तुम्हाला सांगतो आता इथे इमोशनल होण्यासारखं नाहीये. आपण शांतपणे तिकडे घेऊन मी पण बघणार. हाय गणेश.
Hello sir, good evening.
गुड इव्हनिंग. गुड इव्हनिंग.
काय करता तुम्ही?
काही नाही, मी सगळे आता पेशंट थांबवले. दुसरं काय आज संध्याकाळी नो अपॉइंटमेंट्स केलंय.
Something interesting. आता बघूया. We have all good speakers.
मी आता भालूला तेच म्हणत होतो कश्यपे डॉक्टरांना की म्हटलं बघ काय टॉपिकच समजत नाही तर आपलं ऐकावं.
Good evening.
Hi, Ganesh.
So we have good speakers and we have coupled them up with more good people. So we get good insight or light insight into a deeper topic. We are already live.
Yes, we are. Can you hear me?
Yes, I can hear. I mean, so whatever. Once our people join, I put a message to everybody actually.
এটা খুলে নিয়ে আয় তো টেবিল ল্যাম্পটা করতে খুলে ওদিকে বল
हा आवाज ऐकू येतोय ना? मला
Spoke to. So we'll start with transcriptomics.
Okay. After that, what? Transcriptomics?
Proteomics. Then
Okay.
metabolomics. Then radiomics and last microbiomics. We'll change the order a little for speakers' convenience and first three for our convenience.
Okay.
आवाज नहीं clear.
घंटा पड़े
hello. Ganesh, can you hear me?
I can hear you now, sir.
Hi, Ganesh ji.
Hi. Good you joined. Thanks. You're not operating today. At this time.
Nephrectomy is going on, but registrars are doing it, so I'm all right.
That is good. Telemedicine.
I'm sitting in the coffee room.
Hi, Gagan. Hi. We still have the speakers to join.
Yeah.
I have called
Hi,
everybody.
Gagan, good evening.
गया से जाएगा नहीं
हा आता लगेचच याला पाठवू
अनप्लग नहीं है पुढे आज बंद तुम तो सॉरी रे
लेफ्ट विनल कोसामध्ये आहे अप्पर ॲबडोमेन कशाला कशाबद्दल बोलतोय इडियट चला सिटी चेस्ट होतो ठीक आहे
आपल्याला कळलं ते बरंच झालं ना.
याला पाठव आणि आत्ताच बोलून घे काय तो काय नंतर सांगतो म्हणाला का आयसीयू
Hi Ganesh, hi. I'm stuck on the traffic here, so.
Oh my God.
बहुत traffic
That's
है.
quite bad.
Anyway, I left TMS at अभी तो वाशी ब्रिज पे है शायद. I hope talk के time bridge cross हो गया होगा. Anyway, mine is third, no?
Metabolomics. Yes, metabolomics is third. एक विक्रम से बात एक बार फोन पे बात कर लो ना विक्रम से.
मैं कर ले.
I think Sharat also should join. I'll call him.
लंबा दे
No problem. चलो all okay, all the best. कितने लोग हैं?
One minute. I have something to do.
But I think
20.
fantastic, innovative program. Yeah, it will be, hopefully we'll
Let's hope now. The intent is the experiment is for that.
Chalo.
Full credit
Yeah.
to
Chalo,
Ganesh
I'll,
for coming up with such an innovative program. Very, very, very different.
Hi, Raghavan.
Hi, Vedant. Hi.
Hello, Amit.
Hello. Who is that? Can't hear. Hello.
Amit, I think we should start. It's time, 7:15.
Yes, but people were got the notice.
No, we don't. USI program don't wait for people to log in. It starts on time.
Okay.
First speaker is, I just spoke to him. He's having something he's joining, but they had some problem joining. So we'll wait for the speaker at least.
Yeah.
Okay, Ganesh.
Okay. So good evening, everybody, and welcome to this Omics in Uro-Oncology Peep into the Future. We thought having a topic like this, which would probably dealing with basics of some things like what goes into omics, would be a good change for everybody rather than the normal prostate or the renal cancer which goes on and on. A lot of meetings have happened in COVID, and maybe these are the some things which are coming up in future. Till when I was small, omics was only comics for me. Then it was gastronomics, and hopefully today we get some small perspectives into what omics. Omics is basically a collection of scientific biological data in large numbers and the study of that to tell us into various branches of omics as to what this kind of science can tell us about diseases, and particularly we want to go ahead with these omics in uro-oncology. So we have actually 5 good speakers today, and the topic is even difficult for myself also. So I have got 5 good coupled discussants with 5 speakers, and each is going to be a team on the particular topic. So after the presentation basics, I would hand over each to the individual discussant, and we do have a time limit, 10 plus 46 minutes for the discussion. And let's go on with it. The first speaker, though we had a certain order in the brochure, we are going slightly by a kind of a scientific order now. So we would begin with transcriptomics, and the speaker is Dr. Sharat Chandra. He is professor of research at ACTREC, Tata Memorial Center, and a scientist. And the discussant with him is Dr. Raghavan, who is our uro-oncologist at Chennai Apollo. If Dr. Sharath has joined, then please go ahead, Dr. Sharath.
Yeah.
Raghavan, you are actually the session moderator for that.
Sure. Thank you, Ganesh. It'll be a complete pleasure and honor to introduce Dr. Sharath. Dr. Sharath is in Tata. I spoke with him a few days ago, and I was very impressed with the works that are being going on there. So I am equally looking forward to listen to the starting of the omics now. Let's hand it over to Dr. Sharath now. I have a lot of questions to ask him as well. Let's hear from him first.
Thank you very much. Can you see, guys? Can you see my desktop?
Yes.
We can see your wonderful face at this point of time.
We can see you, but we cannot see your screen actually.
Yeah, that's what I'm trying to say. Whether I have the rights to share the screen or not, I don't know.
I think you will have, because I think it's like
Sir,
everybody.
you are the co-host, so you have the rights. Sir.
Acha, Sharath, you are the co-host, right? You have the rights.
Yeah, I'm just trying to share the screen, but it's the options are not available.
Will be in the bottom.
Options
Yeah,
are
I
in the
know.
bottom actually.
But it's only who can share that. That's the only thing it's coming now. Strange. I don't know. I can't share the screen. That's strange.
Dr. Sharath, you have the full bottom showing your mic, video, and everything, right?
Yes, I can see the options, but here only who can share. Advanced sharing options. It's coming on all participant and all participants. There is nothing else is coming. Actually, there should be an option that sharing screen.
So all
why don't you log off and rejoin?
Maybe that's the. Yeah, I will do that. Just give me. Sorry for the this.
Raghavan, maybe you can give some prelude to your topic of transcriptomics.
I can give some prelude to transcriptomics. Can you all hear what I'm going to say?
Yeah,
First and foremost, I am learning in this process. So thanks to Ganesh and Dr. Sharath in this. So my research work used to be in environmental carcinogenesis on prostate cancer, at which time I used to spend 2 years in the laboratory trying to isolate the RNA, the most elusive RNAs that keep running away all the time, and then try to understand whether the expression panels are different. So that was my experience of it. We used to collect the tissue, powder it, and then take it from there. But now things have changed a lot. We all know that DNA is the baseline. From DNA, we have the RNA, which is transcripted out, and the transcription process, out of the RNA, only 5% or 10% of the RNA gets transcripted eventually. That transcription is what is being captured in the process of transcriptomics, is my understanding of it. So nowadays, everything has changed a lot. Previously, each expression molecule has to be measured by a separate RT-PCR test. But nowadays we have what we call gene panels. So the gene expression panel, which contains the measurement that we want to measure, or the RNAs which we want to measure, or the transcripts we want to measure, are pre-done, and then it is available now. These are my understanding of it. I did some more research to see whether where is it used in the prostate cancer spectrum. I couldn't get much of details on it. So I asked Sharath whether he's using it, and he said to me that with the help of Ganesh and Gagan, they are doing some research work on the uro-oncology spectrum as well in the transcriptomics. Whether it is translated into the clinical use or not, I don't know. I said, not to my knowledge, but these are the things which are going to become in clinical use in the next, in my anticipation, 5 years time. Nowadays, most of the genetic panels are now available for us to use in clinical basis. So it is only a question of time when all these things also will become available for the clinical use for our patients. My guesswork is that it is going to be predominantly used in the treatment spectrum, where we will be assessing the validity as whether a particular gene expression is going on for us to use a particular medication. So the medical oncology friends and forums will be much more keen on this aspect of it. In terms of surgeons, how far this will translate into surgical use, time has to see whether it will translate, in real-time surgical use. Those are my understanding of it. Anything else I can highlight more from what I did the reading for the last 5 days. Ganesh, I can't hear you. Ganesh, you're muted.
So, Dr. Sharath, are you able to share screen? I mean, you are muted. We can't hear you. We can't hear you actually. If you are not able to join, then we'll call the next speaker and the team. Like Dr.
I have some technical glitch here.
I think let's not waste time and let's call Dr. Puneet Aluwalia, who's the next speaker for proteomics, along with Dr. Bhalchandra Kashyapi. I think Sharath, you solve your problems, and if possible,
Thank
you
you.
please connect to Dr. Raghavan and be ready for the next talk. Or let us know here that you have not been able to. Please. Yeah, I think I welcome Dr. Puneet, who is a professor of uro-oncology at Max Group of Hospitals, New Delhi, and Dr. Bhalchandra Kashyapi, sir, who is a uro-oncologist at various hospitals in Pune, Jahangir, Dinanath, and many more. And over to him for the session. Dr. Puneet, please share your
It
screen.
needs to be unmuted.
So the team
Yeah,
is...
I am unmute now.
Thanks.
So,
Go ahead.
yeah.
Good evening.
Thank you so much, Dr. Ganesh, and thank you, USA, for inviting me for this talk on proteomics in uro-oncology. And I've been in touch with Dr. Kashyapi regarding the same, because it's an interesting topic, but nevertheless, it is not an easy one to speak on. So I'll just share my screen. I think this slide set is not open. 1 second. Just give me a second.
You want me to share from this side?
Can you see my slides?
No.
No, but I'm pressing on sharing, but it is not showing that open file of my PPT. Yeah, now I think now I got it. 1 second.
Yes.
Yeah,
We
1
can
second.
see you.
Yeah, now it's okay, I think. So let me just start on with that. So Dr. Raghavan was just talking about the RNAs and the genes about it, but we know that the DNA is the blueprint of our human biology, but it is the proteins, it is the signal transduction downstream, which is actually the more important thing, because the signal transduction pathways and the proteins or the enzymes, which when get altered, these lead to altered cell functions, and then cancers develop because these pathways are altered. So these pathways, if you carefully ever see the leaflet of any drug company, like I saw the other day regarding everolimus, they talk about the mechanism of action is all about which receptors or which enzymes they are going to block. It is not about the gene. They mention a passing reference about the genes, but it is all about the final proteins. So next generation sequencing can help us identify the mutations, but it provides little information about the functional consequences of that mutation. Hence, there is a questionable utility of genomic based approach. And everyone knows James Watson. He is spent his life studying DNA. He studied the double helical structure of DNA. He said in 2016 that locating the genes that cause cancer has been remarkably unhelpful, and the belief that sequencing your DNA is going to extend your life is a cruel illusion. This is because once you may have a same diagnosis, two different patients may have a same diagnosis, but the development of cancer is altogether different because of the different pathways, and that is why a particular drug may work in one patient but may not work in another. And also a particular a particular patient with a similar diagnosis may behave very differently, because you are going to have a different subset of populations which in some of patients may actually respond to a particular drug, but some may only have the toxic side effects of the drugs and not respond to it at all. So hence there is a need for giving a personalized care and particular drug to a particular patient or particular treatment to a particular patient, and that can only be done if you study the end point of the thing, if why the cancers are developing now and how they are behaving in a particular patient, and that is all about proteomics. So proteomics is a study of a proteome, which is defined as a complete set of proteins in the cell, tissue, or organism at a certain point of time. Mind you, it is a highly dynamic in nature, and that is why this is the first limitation in studying the proteomics. What I'm going to give you today in my talk is the overview of proteomics, why it is needed, what are the limitations, what are the drawbacks, why we cannot still apply it in our clinical practice, and how we look forward to overcome these limitations in order to get those important biomarkers. So this is the first limitation. Now, for proteomics, proteomics means you have to have some biomarkers, which these are the end results of proteomics. These biomarkers can help you in identifying the disease early, and not only that, it can also help you if you study a particular patient with RCC, for example, which I'm going to give you an example later on. So it can also tell you which biomarker is relevant to which stage, or which biomarker is relevant to which grade. That means that if a particular biomarker is present, the patient is going to behave in a particular way because it is related to that stage or that grade, and hence it can also tell you about the response to the treatment, and also whether that cancer is going to progress or recurrent. So for example, in kidneys, we don't know still whether a 2 centimeter tumor is a oncocytoma or it's a RCC. So if you have biomarkers well developed which can identify that before surgery, you can just avoid the surgery in that patient. So to understand proteomics, you have to actually understand the workflow algorithm. I'll just give you a brief overview and the basic of it, because this is important, because unless you understand this, you will not know what are the limitations and why we are not able to develop biomarkers. And even if they are developed, when you start using it in clinical practice, which may be years from now on, so you have to have these things in mind to understand. You cannot just totally rely on them. So to start with proteomics, you need to have first a sample, which can be either a tumor tissue sample or a plasma sample from a patient with a control, or from two different tumors, or from benign or malignant tumors. And if you're this is again a limitation, because having a good quality and good quantity sample is a problem, because if you use tissue samples, these are usually fallen in pricks and has been used for HNE and IHC staining, and nobody has ever thought of proteomics, so you don't have that much tissue. If you use plasma samples, then these are actually containing a lot of albumin and transferrin, and so you have to immunodeplete these samples so that you get only the proteins of interest which you need to study. So getting a good quality and quantity of sample has been a limitation, and it is still not, we have not been able to overcome this. Once you have that good quality and quantity sample, you subject it to mass spectroscopy, which is a very important component of proteomics. And mass spectroscopy analyzes these samples and analyzes different proteins or peptides in our cases differently, and produces different spectra, and tells you the minute differences, what is present in differently in a cancerous patient as compared to the normal patient. And ultimately you develop these biomarkers which is present in tumor and blood plasma and not in the normal tissue, and then these biomarkers need to be cross validated with Western blot or ELISA, and then only can be used. So there are a lot of steps in which we are lagging behind, and still we need to overcome these. So, say for example, mass, this is a generalized mass spectroscopy framework. You need a sample, which I told you what are the limitations in that, and then you that sample has to be, you cannot just keep a sample there and it will just analyze it and give you the biomarker. You have to have that sample in ionic form in a gaseous phase, because this mass analyzer works in very high vacuum, and then it analyzes it, and when a particular peptide of particular mass travels through this and hits the detector, then detector detects different peptides differently, because depending upon the mass and the time of flight, which the time it takes to pass through that, and gives us a spectra which can be then visualized with digitizer. Now I'm not going to there are multiple different ways of standardizing this mass spectrometer, because there's a technique in insert here, MELD, which is metric associated for ionizing the specimen. There are multiple techniques, all are not standardized yet. There is some limitation or the other with each one, and that is why the end results are not that good. Even if you have a detector which gets depleted, the plates of detector get depleted, the intensity of the signal goes down, so the spectra amplitude goes down, so the end results are skewed. So if somebody changes that detector in between, then your results get skewed, because the intensity will change. You cannot compare, and over time there is a lot of things needs to be standardized in these. So this is the time of flight, which I was saying, that mass spectrometer, the particle goes from here to detector plate, and depending upon the mass and charge of that particle, different peptides are identified differently. So now, with this basic information, we have studied a lot of patients in different urological cancers. Like for renal cancers, different studies have shown there are different markers, like these are the proteomics involved, so people have identified. So like I said, there is no particular marker which I can tell you right now that this marker is for particular this stage or this diagnosis or this grade, or this patient will behave differently like at this point of time. But this is what is going on right now. So there have been studies which have tried to identify markers for the proper diagnosis of RCC versus oncocytoma. If a particular marker is present, these grades and the stages of tumor can be differentiated, and then you can actually, like in studies we are now thinking of giving adjuvant therapy of suppose pembrolizumab or so on and so forth for the patients with higher risk tumors. So if biomarkers are present, they can better tell you better stratify higher risk tumors. Similarly, prognosis can be studied. For prostate cancer, a lot needs to be answered as of now. PSA is the only marker which we have, but there are a lot of questions: which patient needs to be biopsied or re-biopsied, or which patient needs treatment, or which patient will actually benefit from the treatment. And we have many gene-based panels which try to answer these questions, such as 4K score, ProMark, Chlorarrays. Most of these are gene-based, but 4K and ProMark are protein-based panels. And one more thing which I want to tell you is that it is the panel of biomarkers. It's not a single biomarker which will be important. So a lot of protein needs to be studied. So it makes the process all the more cumbersome. So yeah, unless we have standardized everything, it will not be an easy thing to go on. So now you have seen that last slide which showed that gene-based panels which are available right now, or this is proteomics based marker which have been evaluated. And if different studies have evaluated different markers, I have gone through around 8 to 10 papers on each cancers, and I could not, what I tried to correlate was whether these markers have been repeated in different studies, and many of the studies had different markers altogether. So it is the process and the concept which is important right now, rather than having a biomarker. Similarly, CA bladder, urinary based markers are important for diagnosis of non-invasive disease, and tissue based for predicting outcome and treatment response in invasive diseases. Now, again, these studies, it's a congested slide with a lot of studies. In the left column, you can see these many studies have been done with each individual protein or biomarkers like apolipoprotein and all those things. So to conclude, it's a promising approach, can help in early cancer detection and prognosis and prediction of treatment. However, biological fluids can also be used for non-invasive strategy, but it is more easier and more accurate to use tissues. And the most important thing is the ability to validate these markers through the use of standardized workflows, and multi-center participation will be vital. And then use of next generation MS instrument will play a key role, and it will be a panel of biomarkers rather than a single biomarker. Thank you.
Thank you, Puneet. Wonderful. You went through a lot of literature and you have come up with these slides. So, a few points I would want to share. Proteomics will come in clinical practice in next few years, maybe 5 years, maybe 10 years. But as Puneet mentioned, there needs to be a lot of standardization and there needs to be validation of various markers. Looking at the number of markers in bladder cancer or prostate cancer, we may need to focus on most promising markers, and more important, how they can be used easily in the clinical practice. One of the limitation is going to be the availability and the cost also of these markers. But no doubt, there are a lot of unmet clinical needs where we can't answer some of the questions. Who will respond, who will not respond? When, for example, prostate, as you mentioned, when to biopsy, when not to do a repeat biopsy, bladder cancers, prognosis. So this is an exciting field which will develop in future. Perhaps we as clinicians should help in encouraging such studies and get our clinical scientists, and we have people from ACTRIC here, from Tata Memorial Hospital. We should be able to do more studies, and maybe few of the scientists can get together and narrow down few of the most promising markers based on the technique, the ease of applicability, how it can be transmitted everywhere. That is what I feel would be of proteomics, and a single marker versus a panel of markers. That is what would be perhaps more important than relying only on a single marker. Thank you, Puneet.
Thank you so much, sir.
Thank you, Puneet, and thank you, Dr. Kashyapi, sir. I think we'll go to the previous one. Dr. Sharath is back again. Are you able to share now? Oh, excellent. Thank you. We are seeing your screen. So, Raghavan, you are there, right?
Yes, sir. Very much here, sir.
Sharath, you go on. Thanks.
Sharath, please start.
Okay, sorry. Hello, can you hear me?
Yes.
Yeah, we can hear you, Sharath.
Yes.
Please carry
on. Okay. So basically, my lab focuses on the tumor microenvironment, and we use routinely the transcriptomics in the lab to study the different type of the lung cancer and pancreatic cancer patient-derived samples, as well as the cell lines. And Okay. So basically, in the transcriptomics is a study where we try to analyze the total mRNA from a given cell. So what is mRNA? The genetic information encoded in a DNA, that is, all genes are present on the DNA. The DNA, the genes are copied to the mRNA, which is nothing but a transcript, and the total mRNA from a given cell called a transcriptome. Okay. So the entire mRNA or RNA, entire RNA from a given cell is called transcriptome. Okay. So this is the analysis, the study of the transcriptome is called transcriptomics. Ideally, this definition is transcriptomics is the study of total mRNA molecules, that expressed by a particular cell type of an individual. Okay. So why we have to study the transcriptome? Because if you look at any cell, the total number, the DNA is a same. Okay, the genetic information, if you look at the genetic composition of the any given cell, like epithelial cell, fibroblast cell, kidney cell, or prostate cell, the genetic composition is same. But the cell expresses a different set of mRNAs. So only few set of genes are expressed in a given cell type. So that the genes are expressed in the form of a transcript, that is mRNA, or miRNA, or different types of RNAs. So that is the RNA composition defines a given cell type. So if we study the RNA composition of a given cell, we can identify what kind of a cell type is. So here in the transcriptomics, basically, as I mentioned that it includes all types of transcripts, including the messenger RNAs, which are the RNAs they are translated into the protein, and there are some non-coding RNAs, includes microRNAs, which are small 22 nucleotides length, and there are other small RNAs like piRNAs, snow RNAs, and these are the different types of RNAs which are very minor portion, but however in many cancers these RNAs are also differentially expressed. There is another class of RNA which is called long non-coding RNAs. The long non-coding RNAs are nothing but they do not code any protein, but however they have very functional significance in the normal as well as the cancer tissue. Okay, so basically in the transcriptomics we study the entire set of RNAs in a given cell, whether it is a normal tissue or whether it is a cancer tissue. So basically in the cancer biology we try to understand what are the differentially expressed mRNAs or miRNAs in a normal versus a cancer tissue. Okay, so how to study the transcriptome? So basically there are different methodologies or different techniques are available in the literature. So in the early 2000, the microRNA is a very famous technology, and then later on the advanced technology like RNA sequencing. So these are the two technologies. Are the very robust technologies, and there are some other technologies like SAGE, serial analysis of gene expression, and qRT-PCR. So these techniques we can study the transcripts of a given tumor tissue or any other disease tissue, only few genes, okay, for few genes. So I will try to focus in this presentation mostly on the microarray, that is and RNA sequencing. These are the two robust technologies are widely used in the cancer biology. And qRT-PCR also can be used, but we cannot study many genes at a given time point. Okay, we can maximally study I think more 20 to 30 genes at given time point. Okay, so so I want to introduce one more term that is called reverse transcription, which is very important and relevant for this presentation. What is reverse transcription? In the transcription, the DNA is copied to the mRNA. Okay, so this is we call transcription. In the reverse transcription, what happens? The RNA will be converted to the cDNA. Okay, by called reverse transcription by a enzyme which is basically isolated from a virus. Okay, this enzyme converts the RNA to the cDNA. So this process is called reverse transcription. As you can see here, we can see that in this process the mRNA molecule is converted to the cDNA. Okay, so this is one of the very important process in the RNA sequencing or the microRNA technologies everywhere. The reverse transcription, one of the very important process. Okay, so now if you look at how do we study the transcriptome of the given cell, is that by using a robust technology like a microarray or RNA sequencing. So if you look at the microarray in the early 2000 to 2010, a lot of microRNA was widely used across the different types of cancers. Okay, so what is microarray? Microarray is nothing but a glass slide where the small probes, like this is a DNA molecule per given gene. So when you hybridize with a mRNA, it will go and bind to the complementary sequence, and then you can see the signal. So here you see that this is a RNA. So RNA is converted to the cDNA, and which is fluorescently tagged with a fluorescent probe, and now you hybridize with a basically this silicon chip. Okay, so where the DNA probes are there. So if at all your mRNA goes and bind to this particular one, you see it like this. So then the signal you can read by the machine. Okay, at the end of the experiment, you can see in terms of the heat map, you can also convert into the values. Okay, So basically, the microRNA technology will give you, you can study the normal versus a tumor tissue. You can take a normal tissue and tumor tissue and label with the different probes like Psi3 or Psi5, which is a basically green fluorescent probe and red fluorescent probe. And then you can see the comparative expression of a given tissue. Okay. So this was widely used in the, across the different cancers, breast cancer, prostate cancer, many other urological cancers also. I will give you specific examples in my, later presentation. So the latest, so what is the limitation of the microRNA technology? It is not a quantitative. Actually, it's a more qualitative technology. And at given time point, you can do only certain number of genes. So if you take a tumor tissue, I think more than 10,000 genes you cannot do for this microRNA technology. But with the latest technologies like RNA sequencing, if you look at that, RNA sequencing, you can do many genes you can study at given time point. Okay, so how do we do the RNA sequencing? So basically, this is a sequencing method. As name itself, it says that we convert we take the tumor tissue, isolate the RNA, and eliminate the ribosomal RNA and tRNA, which is least interesting for us, and you enrich for the mRNA by using a poly A tails. So the mRNA has a poly A tail, and we use the oligo-DT beads. So basically, by using oligo-DT beads, all mRNA will go and bind to these beads, and then you can only have the mRNA. So then you convert this, make the mRNA fragments and do the reverse transcription. As I mentioned that in the by reverse transcription, you are converting the mRNA into the complementary DNA. Okay. So these complementary DNA, further you make the ligation, adapter ligation. The adapters are nothing but small molecules which you ligate to these small fragments, and then further you do one round of PCR, that is to amplify the molecules, and then we do the high throughput sequencing. So basically here what we are doing, we are taking the mRNA and we are converting to the cDNA, and we are doing some process, and then we are doing a sequencing. So in the sequencing, what do we get? A raw data. By this raw data is converted to this, bioinformatic analysis. You get the sequences like this. So this is here. You get every single mRNA sequence. Whatever the cell expresses, every single mRNA sequence is, you can get the information and the quantity also. So you will get the absolute quantification of a particular RNA. So what are the advantages of RNA sequencing is that, if you look at the normal versus tumor tissue, you get the absolute quantification, as well as you can identify the novel molecules. Microarray, you have to have a probes that are very specific. Okay, if you have 800 gene probes, that means you can only identify the 800 gene expression only. Okay, but here in the RNA sequence, every individual mRNA can be sequenced. There is nothing is like that. Okay, there is no bias. Okay, and also it is very quantitative. Okay, so if you look at the this is a classically, the differentially expressed mRNAs. This is versus a normal tissue. You can see, this is the heat maps. Okay, in the heat maps, each line is represents one single gene or one single mRNA. Okay, the red is in terms of the high expression, and the blue you can see the lower expression. Okay, so these are basically this heat map was taken from one of the published paper. So you can see that these are the duplicate samples. You can see the expression is very similar, and these are the different samples. Okay, these are the normal fibroblasts. Here we have taken the cancer associated fibroblasts which are present in the stroma. Okay, including the prostate cancer also, this stroma is very high. So we are trying to understand what are the differentially expressed, genes, mRNAs between the normal fibroblasts versus the cancer associated fibroblasts. So this is the typical way of representing the differential expressed genes. So what we do further, we do validate the sum of the genes and which are differential across the different tumor tissues or different patient samples, and then people will come to a signature gene, that this is a signature gene expressed in the given particular cancer type. Okay, so this way people do generate the signature the signature genes for the prognosis or the the disease progression or different type of the purposes. All right. So this is this is all these methods are for the bulk
Sharath,
sequence. Yes,
Dr. Sharath, this is a 10-minute talk, so
Sorry.
I would request you to
Okay,
just come
sure.
to the clinical use,
Sure,
so
sure,
that in
sure.
another 1 or 2 minutes we can finish that.
Sure. All right. Okay. So just one quick thing. Single cell transcriptomics is nothing but to identify the total total mRNA from the one single cell. Okay. So this is the latest advanced technology where you can see identify the different types of the cell types in a given tumor tissue, whether it is a prostate cancer or that breast cancer and all these things. So what we see here is that if at all there is a one particular type of a cancer cells, then we can identify this is we can relate with the poor prognosis or the disease progression. Okay, so what are the benefits? These are the some of the commercialized prognostic gene expression signature diagnostics test for the FDA approved. So here you can see for the prostate cancer. So this is the one for the here they have used the 31 gene signature for the FFP samples, and this is routinely used in the in abroad. Okay, this is one of the specific example I have taken from the 2018 paper. The 17 gene assay to predict the prostate cancer aggressiveness in the prostate cancer. Okay, and so these are so basically people have done the RNA sequencing, and then they have identified the certain genes which are differentially expressed between the the aggressive versus the less aggressive patient samples, and then they have made the 70 gene signature. And another paper also analytical validation of the Oncotype DX prostate cancer assay. So this paper I have seen in the many in the clinics also, this 12 gene the 12 cancer related genes the signature has been used, and here they have used the these are the 12 genes, and they have used the normal genes. These are the reference genes, they are the control genes, and they have taken that when they have these 12 genes, they have done the expression analysis by using a RT-PCR that is very well known during the COVID the
Sharath, just moving forward, just 1 point or 2 points about the clinical validity, especially what do you think is going to be the situation in the next 5 years? I was telling the audience that in the next 5 years, we'll anticipate at least some clinical validity use. Oncotype DX is already available in clinical use. Do you think this will be extended to the other tumor types as well, like kidneys and bladders as well?
I think his window is frozen, so looks like his link has cut off. I think most of his discussion was done, right?
Yeah, it is done. He was into
So
the clinical use. So answer to the question, I mean, waiting for Sharath to come. Answer to the question is that I anticipate at least prostate cancer related gene expression panels will be widely available even in India in the next 5 years' time. That is my anticipation. But in terms of such a panel is being developed by kidney cancers. So in the next 5 to 10 years, we can start expecting the kidney cancer panel also coming through that. Regarding bladder and other cancers, I think they are some way yet to go. Prostate and following that, kidney will come through. And I think with that, we will finish this talk, Ganesh.
Okay, I think that was still a nice talk. I mean, we got to know the basics of transcriptomics, the mRNAs basically, and your comments were very valuable. I invite the next speaker, Dr. Vikram Gota. Good evening, Vikram, and thanks for agreeing to be here. Vikram is a very renowned person. He's a professor of research at a renowned Actrix Center, and coupled with him is another renowned person, Dr. Vedang Murthy, a very good personal friend, professor in radiation oncology at Tata Memorial Center as well as Actrix and TMC, and a lot of other activities. Vedang, very premier. I hope you have.
Thank you, Ganesh. Yes. Reach Belapur. Yes.
Thank you, Vedang.
So thanks, Ganesh. We will talk while Vikram is loading up, metabolomics. Vikram, you are ready to talk? We can't hear you. At least I can't hear you. So moving from mRNA and proteins, we are going now to the metabolites of the body, essentially the study of metabolites in the body fluids and tissues. So that is what metabolomics is. And let's see what the role is in urological cancers.
So, yeah, I think we can see the first slide, but we cannot hear you yet.
Yeah, Vikram, please speak. Vikram, you're there?
The voice is not so audible, but we can hear you like.
Hey Vikram, what are you doing?
No, there was some problem. Now it got sorted. I mean, I was not allowed to unmute. Now it is sorted. Yeah.
Okay, thank you.
Okay, okay. Thank you very much, Dr. Bakshi, for this kind invitation, and thanks Vedang for introducing metabolomics. As Vedang rightly said, metabolomics is the study of the metabolome. Oh my God, I'm not able to move my slides. So what happened? Yeah. So I'm going to talk about the following items. One is I'm going to introduce what are metabolites, the examples of metabolites in clinics, what is metabolomics, applications of metabolomics, the metabolomics workflow, and its applications in uro-oncology. So now, I mean, everything in the body, has to be formed or broken down through metabolism. Okay, so metabolism happens. I mean, you name anything, like you name a protein, you name a lipid, you name a carbohydrate. So everything is a product of metabolism. Either it is anabolism, which is kind of a building process, or catabolism, which is a breaking down process. So now there are types of different types of metabolites. Most importantly, the primary metabolites, you could call them endogenous because you have these carbohydrates, proteins, nucleic acids, lipids, which are present in the body all the time. And there could be secondary metabolites, which are a product of xenobiotic metabolism. Xenobiotics are those which we consume, like it could be antibiotics, it could be pigment, it could be toxins, whatever. So the products of their metabolism are secondary metabolites. Now, just to give you some examples of metabolites, sugars, ketones. I think we discussed this in the previous slide. And now the sum total of all the metabolites in the body is the metabolome. So, and the metabolome is a very dynamic thing. I mean, in the sense, it may be different from morning to afternoon to night, depending on your mood, your stress, and then external stimulus like atmospheric pollutants. Different diseases like a common cold can affect the metabolome in different way. Cancer can affect the metabolome in different ways. So that is why metabolome may have a role in, let's say, since we're talking about cancer, it may have a role in cancer diagnosis and then prognostication and so on and so forth. But these are the various things, including nutrition, which can affect your metabolome. Okay. So now, when you talk about metabolome and metabolomics, okay, there are two different ways in which you can do metabolomics. Now, just imagine that you are going on a fishing expedition, okay, and not knowing what kind of whether you'll catch a fish at all, what kind of fish you'll catch, and so on. So that is called as an untargeted metabolomics, okay, wherein you kind of look at all the metabolome present, in the matrix. By matrix, I mean it could be blood or saliva or urine or tissue or whatever. You look at all the metabolites and then try to discriminate between groups. Like for example, you're looking at prostate cancer versus non-prostate cancer groups. You look at untargeted metabolite in the urine, metabolome in the urine, and then see whether there are any relative differences in the peaks of certain metabolites, the two sets of patients or cohort. So that is untargeted. Whereas on the other hand, you have a targeted metabolomic analysis where you know what exactly you're looking for. And that information can come from, let's say, proteomic studies or genomic studies and so on. So in the proteomics and genomic studies, let's say it has shown that there are certain pathways which are getting affected in a given disease. So then you know what exactly are the products of those pathways and you know what to look for. So when you know what you're looking for, That is called as targeted metabolomics. So just to give you a sense of how they kind of stand with respect to each other, imagine a quadrangle. Okay, you have a quadrangle here, and then you have non-targeted on one side, you have targeted on the other. And non-targeted is very high on data complexity and variability because you are looking at and the whole range of metabolites. So it is a very complex data, and also obviously the amount of information that's available is extensive. On the other hand, targeted is very high on stability and repeatability because you know what exactly you're looking at. So it is definitely going to be stable and repeatable, and you're able to quantify it. Like as you can see, I mean, it is useful for quantification. Whereas non-targeted metabolomics is mostly a qualitative analysis. Okay, and it is also, so quantitative accuracy is very high with targeted metabolomics. And this is the workflow wherein you have a biological sample, let's say blood, you run it through mass spectrometry. Mostly mass spectrometers are used for metabolomics. And then you extract data from the mass spec. So in mass spec, what do you say? It will throw out a series of peaks. You'll get, when you run a plasma sample, you get about hundreds and thousands of peaks. So now how to make sense of that? So you have to correct for the baseline, you have to deconvolute it, there may be a lot of artifacts. So trained mass spectrometrist will be able to differentiate a real peak from an artifact. So you have to do the differentiation and all those things, and then clean the data basically. Once you clean the data, it is now ready for statistical analysis to see whether there are any differences between groups or not. And then you will also be able to identify the compound based on the charge to mass ratio and so on and so forth. And through a combination of compound identification and statistical analysis, you will be able to arrive at certain interpretations. Okay, so this is in a nutshell the metabolomics workflow, and applications are plenty. So in human disease, you can use for diagnostic and then biomarker discovery and risk determination, prognostication of disease. In drug discovery, you can, early in the drug discovery, you can use metabolomics to see how things are getting altered and whether those alterations are going in the right direction and in the direction that you want. Okay, so that you can, whether you should continue with the drug or not. And then toxicology, definitely. And then any discovery science, I mean, you can use it because, anything you give from outside is going to change your metabolism, metabolome. And that way you'll be able to find out whether, the movement is the right direction or not. And based on that, you can, either enzyme or drug is working or not working, or working in a desirable fashion or not working in a desirable fashion. So now that is about metabolomics. I'll just give you 3 examples depending on availability of time. And the moderator can stop me if I'm running short of time, so that I always cut down on the examples. So this is one example of, to differentiate between a prostate cancer and a non-prostate cancer, a biopsy negative person versus a person who was diagnosed with prostate cancer. So we have about 62 patients who had who got the diagnosis as positive and 42 who were negative. And then they could identify 21 differential metabolites between these 2 groups. And this analysis is called as a principal component analysis. I mean, let's not go into the details of these things, wherein, you can take a set of metabolites and then, do some complex mathematical modeling and then see whether, you can differentiate between one type over the other. So here the red dots are all the controls and the white dots are the green dots are the prostate cancer patient. So, and then, you can do principal component analysis, component 1, component 2, component 3, where, taking different sets of metabolites. And then you can also see what combination of metabolites will give you the best discrimination between controls and patients. So this is so from this, what emerged was, there are certain, so this is some kind of a grid you form based on what kind of pathways are involved. Then,
Vikram, you have 2 minutes.
Yeah, they could identify that these are related to prostate cancer and these are related to LV, and these are the results from univariate data. So basically, this study showed that you can differentiate between controls and prostate cancer patients based on metabolomics. So I'll skip this and I'll go to third example because that's interesting. So this is diagnosis of bladder cancer and prediction of survival by urinary metabolites. Once again, you take a certain number of patients. So all the metabolomic studies, you will have a training set and a validation set. So basically, you train your mathematical model from the training set, and then you apply that model and validate it in a validation set. So basically, you will get this kind of groups. Okay, if you get this kind of groups, you know that your metabolomic analysis is able to discriminate between those with cancer and those without cancer. And this ROC analysis is also another way of seeing whether what is the discriminated potential of that metabolomic analysis. If the AUC is very high, it means that the metabolome, whatever the sum total of the metabolomes that you are taking, can discriminate between cancer and non-cancer. If the AUC is very low, it means that it cannot discriminate. So this is the general, a study design wherein you had the 250 odd patients. They were divided into training set and validation set, and then you could see that there was a very high sensitivity and specificity by two different analytical, mathematical methods. One is called as orthonormalist partial G-square method, and the other one is receiver operating characteristics, and both of them yielded high sensitivity and specificity. And this one to show that you could differentiate between bladder cancer versus non-bladder cancer, and the blank squares here indicate that patients who had benign hematuria. So it could actually differentiate benign hematuria from actually those who had cancer. And in the bottom panel, it is the metabolome analysis could actually differentiate between muscle invasive bladder cancer versus non-muscle invasive bladder cancer, and this is how the various the univariate analysis looked like. Okay, and then
Thank you.
Metabolites that were picked up, just half a minute, everything. Metabolites that were picked up that could actually predict survival of patients with bladder cancer. So that is one of the applications of metabolomics. So this is in a nutshell, what I can explain in 10 minutes about metabolomics. The applications are plenty, including diagnostics.
Thank you so much, Vikram. Brilliant talk. Thank you so much. I think you did perfect justice to what was asked for. And urology being urology, do you think, so we are always short of tissue, for all the lab work. So do you think urine provides a great opportunity for metabolomics? That is one question. Do you think urine is a great source of, particularly for urological cancers? And second is, what does metabolomics provide that the other guys, the proteomics, and what are the advantages particularly that this provides that others don't? What is the edge that this has that others probably don't? I mean, maybe the two questions from me.
Yeah. So the choice of the specimen is determined by the disease. Like, for example, serum pretty much can be used for almost any this one, because it is a central compartment and every organ will throw their metabolites into blood. So that is one thing. But serum is invasive. So the next step is, are there any non-invasive sample collection techniques? So if it is oral cancer, definitely you can take something like saliva. And likewise, for urological malignancies, urine can be a very good surrogate. And then even from the examples that I have shown you, I mean there are instances in bladder cancer where they actually demonstrated the ability of metabolomics to differentiate between cancer and non-cancer from urinary metabolomic analysis. So urinary metabolome is definitely an option, and especially for urologic malignancies, it should be definitely used. Okay, and
what about the,
the second question is, what advantage does metabolomics provide? See, I mean, we all know that the kind of flow of information. So the flow of information happens from the gene to the mRNA. So we have genomics, and then we have transcriptomics, then mRNA to protein, we have proteomics, and then protein to, the proteins are what? I mean, almost all enzymes are proteins. So as a result of that, from proteins, you have the metabolome. Now, what happens is, one might argue that I'll study genomics because, that will kind of translate into metabolome ultimately. But that may not necessarily happen, because from genome to metabolome, there is a lot of, slip between the cup and the lip, because, there could be various processes like post-translational modification or alternate splicing of proteins, and so many such processes in the body which can actually, not allow the gene to express, the way it has to express. So there could be differential proteins that could be formed from the same gene because of alternate splicing and things like that.
So in other words, are you saying that it is better to study the near end product than the
correct.
Going to the origins of
exactly, it's the most downstream, so that is the advantage that metabolomics offers.
Okay, thank you so much.
There are certain complexities there.
Thank you so much. I think I hand it back to Ganesh now. Thank you so much.
Yeah,
thank you, Vedant, and thank you, Dr. Vikram. It was really nice the way
Dr. Ganesh, can I add one comment? Just one comment regarding
very
that?
quick. We are
Very
late.
yeah. So just to answer Dr. Vedang's question, whether tissue or the urinary or the sample can be used. So there have been multiple studies. Tissue is a better because as I said that these metabolites and proteomes are very dynamic. So at the same point of time, you can take a normal adjacent tissue, which is called as NAT. So you can compare those sideways rather than taking a sample from blood, in which it is difficult to do that.
Okay, thanks, Puneet.
Thank you. Thank you.
And I invite actually the next speaker, Dr. Palak. I hope she's a co-host so that she can share the screen. Palak is Professor Radiology at Tata Memorial Center, and she'll be speaking on radiomics in uro-oncology. And Dr. Amit Ghosh, who is a uro-oncologist at Kolkata, will be the discussant here. So please go ahead.
Yes, thank you so much, Dr. Ganesh. Dr. Amit, would you like to say something, or should I share my screen?
Yeah, please start sharing the screen.
Share the screen. He will end. I mean, he'll speak in the end.
We are sure. Into at least getting onto Mother Earth, because so long we were into stratosphere. So urologists are dumbos, as you know. So we are actually now going to speak on a subject which we are used to, which is the images we are already used to. Now we are looking forward to your talk on analysis of the images and what radiomics has got to do. Offer us.
Yes, thank you so much. So talking of what is radiomics, all of us have been understanding so much about AI, and AI is everywhere. Talking of radiomics from the AI world. My slides are not seen?
No, the slides are not seen at all.
Okay, but I have shared 1 minute. Is it seen now?
It's coming. Yes, yes.
Now it has come. It's just, you go on.
Sure. Yeah, so we move from the world of AI, which is almost ubiquitous and universal to all specialties, coming down to radiology and medical imaging. So radiomics is a method for quantitative description of medical images. So when I say, okay, this is a hyperdense, heterogeneously enhancing mass, or whatever I describe, there is more beyond what I can perceive with my human eye. So for example, if I would have to be interpreting this CT scan for this kidney, I would say it is, I have contrast in the aorta. So this is a post-contrast scan, the kidney scene. I can give certain features of this mass as overall, it is a hypo-enhancing, low-density mass with some internal enhancing septations, and that's about it. I may not confidently be able to say whether this is really cystic or it has a solid component which has a very low density seen on the CT. And then we say that I think I would need to do an ultrasound to characterize this mass. So that is where radiomics steps in, because radiomics is a program which can learn that this is how a cyst will look and this is how a solid tissue will look. So beyond my perception, it is going to help me with certain added features. What do we mean by handcrafting versus machine learning? So when we talk of AI, we have machine learning algorithms. So what it does is, if I was to put that mass through machine learning, the computer is going to generate whatever data it can from that mass and tell me, okay, this is solid, this is cystic, or this is what feature you would want. Versus radiomics is an extension of AI. Radiomics is driven by the data scientist. So if my question is this tumor going to respond to chemotherapy? Does this tumor have, say, 30% of a solid volume? Does it have an aggressive component? So I am acting God in this particular data set. I choose my question. So when the entire study is being done, I'm trying to solve that particular answer for that question. So if my question is going to be, is this going to respond to chemotherapy? I want to see whether there are solid areas which then become necrotic, which is a positive response to therapy. Or I might want to say that, okay, there are peritumoral changes which indicate a response to therapy. So if that is my question, I start looking at these particular answers, which is how radiomics is handcrafted versus just what is generated by the machine. We all need to know what is the term big data. So we have huge amounts of data. So even the CT scans and MRIs and all, we are always crying on uploading scans on the PACS because it is so heavily data driven. But big data is way beyond any amount of data that we are talking of handling in our hospitals. And to process that data, we need a separate set of technology, and that together is called as big data. So the aim of radiomics is to enhance the existing data available to clinicians by means of advanced, non-intuitive mathematical analysis. The concept is to see what is imperceptible by the human eye and what is not accessible through traditional visual inspection of the generated images. By mathematical extraction of the spatial distributions, so signal intensities for MRI and pixel values for MRI, voxel for CT. So there is an interrelationship. From this data, we extract mathematical information, goes through textual information analysis from the field of AI. There are appreciable differences in image intensity, shape, or texture, and all these are quantified together. And this overcomes the subjective nature of image interpretation. As my colleague and I, when we are both sitting, I say, I think this is cystic, and he says, I think this is low enhancing, non-enhancing solid component. That component goes away. So radiomics does not imply any automation of the diagnostic process. Rather, it provides existing ones with additional data. So it is not replacing a radiologist, it is enhancing or empowering a radiologist and definitely giving you standard analysis vis-a-vis the human error can be taken care of. The classification task, this is how the steps are taken. We differentiate what is benign versus malignant, talking about the world of radiomics and oncology. After that, the other common area is for prediction of clinical events such as disease, time of death, or overall survival. The steps are that curation of images taken. First, it has to be the question which is generated by the study design and the study endpoint. It is very important in radiomics. This is not AI. In radiomics, we need to identify what clinical question we want to answer. And then we have a host of other factors that come in. After the curation of images comes tumor segmentation, image pre-processing, feature extraction, model development, validation, guidelines and quality checklists, and digital phantoms. So for example, this is how the steps go in, that there is a clinical data collection. So I have answered my question. I want to look at all cases of multiparametric MRI prostate. So I find a data set of these patients. These patients are anonymized. We have to have our clearances done from the institutes to have anonymized data being transferred. Once the image is transferred in an anonymized fashion, after, of course, the consent and IC approval, images are prepared. By preparing, for CT, we can measure the Hounsfield units, but for MRI and ultrasound, there is no objective measurement. There can be variation of the measuring of signal intensity by pixels or by ultrasound pixels. They look more gray, less gray. So that is heterogeneous. This needs to be first homogenized. Once the images are prepared, then we come to annotation. Annotation can happen either by a radiologist or by somebody else, either manual, semi-automatic, or automatic, depending on who is doing it, and it can either be 2-dimensional or 3-dimensional. 3-dimensional can become consuming a lot of times. However, it depends on the data set that needs to be analyzed. Feature extraction. So now this is done. So there are various features. There are first order and second order features that can be extracted. So first order features would be something like the morphology, volume, dimension, size, intensity. These are primary or the first order features. And the second order features are something like where texture analysis comes in. So let's say, for example, GLCM and all. So that is gray level co-occurrence matrix. So if there are two signal intensities, 2 pixels at a distance of a defined predefined distance, it averages out and says, okay, this is the GLCM. Or if I want to see 2 closely placed signal intensities in 2 pixels, and that comes as a signal zone. So that is a GLSM. So there are various features by which the feature extraction takes place. After that, there is a process of transferring them into bins. So for example, let us say this is an image and I identify the various features in this, they are transformed into bins. So there will be a set of information which is grouped or classified as one particular value. Two intensities which are similar will be grouped as another particular value. These are called as bins. So the lesser bins you have, it's like a dustbin, you're throwing something or dumping into it. The lesser bins you have, it is going to be less noise, but less image information also. At the same time, more bins means you'll have greater resolution and more finer parameters are being considered. Then comes the process of model validation. For validating, ideally, we need about 1/3 of data. So let's say we have 100 scans, there will be 30 cases which will be required for validation. There are different types. I could use a part of these 30 from the 100 and jumble up and put it across the same model that I have been created. Or an ideal situation is to take the 30 cases from an external set. This external set can be intra-institute or extra-institute, and that is what is either internal or external validation. After that, we have the performance evaluation, where again, once your model has been performed, it runs through the entire data set. And of course, there is a new data set which will run through it to look at the validity of the so-called created algorithm. So the utility in urology would be for like kidney, prostate, bladder are the ones which are more explored. There are benign conditions also, such as stones and inflammation and infection, which are there when talking of uro-oncology. Renal masses, there are studies on, say, oncocytomas and small renal masses. For prostate, we have again diagnosis for looking at how aggressive it is on like Gleason score, or bladder for staging for muscle invasive, non-muscle invasive, as well as for prognostication for all of them. Just like 2-3 articles, like for example, this particular article talks of radiomic features measured with multiparametric MRI prostate for detecting aggressiveness, in which they have analyzed identifying Gleason score more than equal to 8 based on the lesion size, PIRAS texture analysis, and histogram. And they have found a very good fair performance of the Gleason score and excellent performance for the Decipher score. This is an example of radiomics in renal cancer, where subtyping and tumor biology of tumors are seen in the pre-treatment phase, and the post-treatment phase can look at prediction of response to systemic therapy, including anti-angiogenic treatment beyond the size criteria. So there is a criteria of what cases are put on surveillance. So the AUA specifies that for renal masses less than 4 centimeters may be put on a surveillance, or 4 to 7 centimeters, depending on the clinical status. But however, a lot of these have, yes, a lot of these have variable heterogeneous appearances, and which is why radiomics is called as a virtual biopsy, because when I sample one area, the radiomics is going to give me an overall perception and aggression of the entire tumor, and not just what is sampled. So it gives us a better overall prediction. And of course, it also helps in predicting for response assessment. Again, bladder, muscle invasive and non-muscle invasive bladder cancer, they have found fairly good results. For example, there was this big study where it had 82% and 81% sensitivity and specificity. And the conclusion was that that it has a fair performance and a good diagnostic accuracy. It also matters, this was a meta-analysis where no matter the approach used for the radiomics, all of them had a good diagnostic accuracy, thereby affirming that, yes, it is a good modality that can be used. Thank you so much.
Thank you, Professor Popat. I think that was an excellent overview of radiomics and how useful it can be. I think we are already used to the advanced sort of imaging in urology and uro-oncology particularly, and you've covered all aspects of it. Radiomics combined with, as I understand it, it's very useful for a finesse of diagnosis. And therefore, combining radiomics with PIRADS, say PSA densities, the mean ADCs, and DRE, this pentafactor actually gives you the insight as to whether it is a clinically significant or a clinically insignificant prostate cancer. So that's one angle. Not only the diagnosis, not only does it give an edge in the diagnosis, I think if you look at prostate cancer, or even before that, even if you look at the partial nephrectomy specimens in a paper that has been recently published, 30% of the partial nephrectomies in America has turned out to be benign. And that is where radiomics is probably going to be extremely useful. if you turn another role of radiomics, which is very encouraging, is that you have very briefly touched on that because of lack of time, you could not. And that is in prostate cancer, there is this FLAME study, which gives focal ablation of prostate cancer, and it gives the dominant intraprostatic lesion radiotherapy, MP-MRI directed radiotherapy. Now, what it had targeted was you give the dominant lesion a real dose of radiotherapy, and you have better results with less side effects. But what it revealed was the dominant lesion may not be the most aggressive lesion. You've touched on it briefly, that there are ways of finding out what is an aggressive lesion, and on that basis, probably maybe on another talk you can elaborate that tumor heterogeneity can be addressed by habitats. And there is something called this habitat risk scoring that is coming in, which is very, very useful for modulating our therapy. But I think we can go on and on. This is the future. And thank you very much for being so comprehensive in so brief a time, and something which we could digest as urologists. Thank you so much.
Thank you,
Thank you
Dr.
so
Amit
much,
Ghosh.
Dr. Amit.
And thank you so much, Dr. Palak. It was nice to hear your talk. So let's go on. We are slightly in a delay because of the login and sharing and all things. So Dr. Srivatsa is the next speaker. Srivatsa, please share your slides so that you get time. And he is going to talk on microbiomics. He is a uro-oncologist from Sri Shankara Cancer Hospital, and Dr. Gagan Prakash, my colleague, ex-colleague and head at Tata Memorial Center, Uro-Oncology Department, would be the discussant here, and Gagan will take care of with Srivatsa for explaining us about microbiome and GU cancers. Thank you, Srivatsa.
Sir, are my slides visible? Am I audible?
Yes. Just.
But I am not able to make it as a PPT format. Just stop and reshare, sir.
No problem. So when I had thought of this idea of having omics, I first shared it with Dr. Amit Ghosh, who was also very much keen. And then we both thought that instead of prostate, we'll change over the topic to omics. And then some of my good friends, Dr. Vedang, Dr. Gagan, we just had a small discussion. I also spoke to one more friend of mine, Dr. Sanjay Adla, and then we thought we should totally change over the meeting to omics. Srivatsa, are you there?
Yeah, I'm there, sir, but I'm not able to make it a full screen because I'm getting this.
So does it play this way?
I think we are all right. Carry on.
We are
yeah.
If you can move the slides, you can play.
Yeah. So very good evening, and many thanks to USI and Ganesh Bakshi sir for this opportunity. I'll be slightly moving away from patient-centric data in advances in diagnosis to something which has been very innate to us, and how this is changing, or this is going to change the way we identify, treat, and manage genitourinary cancers. Slides not moving.
We are seeing it full screen, but
no, but the slide is not moving.
So that might be just internet issue, maybe.
I'm really sorry for this.
No problem. You take time now. We are on the last presentation, but it is still interesting, and we would, as we got other concepts, the whole meeting was to get a preliminary or a very primary concept about this omics in certain ways. So we thought that if things work like this, we could, also focus in detail in one of the things or two of the things, and that would help us spiral the research.
Yeah, I think I'm on now, sir. I would like to thank my fellow Dr. Rahul, who has really helped a lot in collecting a lot of data. So coming to the definition of microbiome, it's all viable microbial genetic material which is available within a niche or within a silo in the human body. The most common nomenclature that will be associated with the microbiome is dysbiosis, which means that which represents the microbial imbalance. And this has actually been linked with genesis and evolution of plethora of diseases, not necessarily cancers, most commonly autoimmune disorders and cancers as well. it is very interesting to know that human microbiome is our second genome. So if there is another set of genome that exists within a human body, it is the microbiome. The microbiota have co-evolved with humans for millions of years and plays a critical role in human health, both good and bad. Effectively, we are only 10% human. The rest of the 90% is a microbial flora as far as the genetic makeup is concerned. So carcinogenic process changes the local microenvironment and creates new niches or alteration of microbial component, and its function contributes to carcinogenesis. This is the fundamental basis on which the studies on microbiome have evolved. Generally, we have to understand that it is not one microbiome leading to one cancer, except gastric cancers, the low-grade maltomas of the stomach, implicated by H. pylori. It is usually an interaction between a plethora of organisms, bacteria, viruses, and fungi that affect either suppression of cancer or accelerate cancer, or for that matter, even alter the way the treatment impacts cancer. So if you look at the cartoon here, several microbial products can alter the immune system and alter the equilibrium between the inflammation and the immune suppression of the normal homeostasis of the human body. Whenever there is a healthy commensal environment, there is a nice equilibrium between the inflammation and the immune suppression within the body. And when there is a dysbiosis induced because of various factors, there is an alteration which can progress onto carcinogenesis. So we have been talking about dysbiosis. What is this dysbiosis and how can we identify it? Is there a methodology to identify it? Yes, there is. I would like to focus on the 16S amplicon sequencing, which was originally done to identify the bacteria, which can also be applied for identifying the viruses, the archaea, as well as the fungi. So this stands as the workhorse of microbiome evaluation in human beings. There are other methodologies by which larger bacteria or larger fungi can be identified using the shotgun metagenome and the metatranscriptome sequencing, but the 16S amplicon sequencing will form the workhorse of identification of the microbiome profile. So has there been any research on microbiota? I was personally surprised to know that the Human Microbiome Project was established in the year 2008 by National Institute of Health, and it has now generated or published the largest microbial genome database for various different body sites, including the oronasal cavity, skin, GI, GU tracts, breast, and so on and so forth. The most extensively studied microbiota-related carcinogenic events are with the intestinal microbiota. Large data exists between colon cancer and the gut microbiome. And it is now known that these intestinal microbiota can significantly impact both in terms of augmenting disease progression as well as inhibiting disease progression in various aspects of cancer development, progression, and therapeutics. For instance, the intestinal microbiota can augment sustained proliferative signaling, enhancing the carcinogenesis. It can also inhibit the natural immune destruction, which happens as a scavenging, thereby again promoting carcinogenesis. It can also augment tumor promoting inflammation. It can induce genomic instability within the host cells, so on and so forth. Coming to the GU cancers as such, there are two distinct entities where the microbiota can have an impact on GU cancers. One directly where the urinary microbiome itself can alter the pathway of carcinogenesis, and the second is the indirect interaction from the gut microbiome, wherein which mostly the therapeutics of GU cancers are going to get altered, which I'm going to elaborate in the subsequent slides. Most of the data from GU is in the domain of bladder cancer. It is now known that the conventional view that urine is typically sterile is wrong. The urine has several plethora of normal commensals in healthy individuals, and modern culture and sequencing techniques have now enabled the detection of these commensal microbes throughout the urinary system. If you look at this cartoon, it is now known that there are a lot of microbes which pervade the healthy bladder, and the differential bacteria which are seen in the setting of a low grade or a non-muscle invasive bladder cancer is quite distinct. In that the bladder with non-muscle invasive tumors are more populated with bacteria like the Pseudomonas, the Fusobacterium, the Acinetobacter, and others. This is the classic example of microbe-induced bladder cancer induction. We all know that Schistosomia hematobicum, but not all patients infested with schistosomiasis eventually end up having cancer. The reason actually is now known that all patients with schistosomiasis will have a release of inflammatory cells. But if the microbiome in the host has only nitrate-reducing bacteria like the Staphylococcus aureus or the Proteus mirabilis, will these inflammatory cells get activated to produce more and more N-nitrozoamines, which in fact are the actual cause of bladder carcinogenesis. So in those patients where these inflammatory cells are not augmented by bacteria which are nitrate reducing, patients will remain to have just schistosomiasis without occurrence of bladder cancer. So what are the factors that affect the urinary microbiome? Almost all known factors like the diet, lifestyle, the genetic makeup of the host, gender, age, interventions, surgical interventions, and the history of antibiotic usage that the host has had can all affect the urinary microbiome. Several studies have shown how the modification of these factors have altered the genera and the species of microbiome in the urinary bladder. It is also now shown in a couple of papers that patients who are smokers, who have a particular set of microbial flora in their bladders, have a higher risk of having an advanced bladder tumor compared to a moderate risk in patients who are non-smokers. Let us come to the the potential interactions of this microbiome and therapy. The classical example has been BCG immunotherapy for non-muscle invasive bladder cancers, wherein we all know that the predominant action of BCG is by its adherence to fibronectin, and it has now been shown that certain bacteria like the Lactobacillus iners can also competitively bind to fibronectin, thereby reducing the efficacy of BCG. There have been several papers where they have tried to replace the Lactobacillus iners by other species of lactobacilli, and these other species of lactobacilli have been shown to synergize with BCG by inhibiting the Lactobacillus iners and thereby augmenting the effect of BCG. The other treatment-related effect comes in immunotherapy. Immunotherapy is now being used more and more in almost all genitourinary cancers. It has been now proven that there is an association between the human gut microbiome and the efficacy of anti-PD-1 therapy. Depending on the gut microbiome flora, there are situations where these favorable microbiota can augment the PD-1 expression as well as response to treatment. There has also been now a study wherein they have manipulated this gut flora with oral agents or fecal microbial transplant of patients who have had a favorable response to immunotherapy priorly, and these studies, small sample studies, have shown that this can actually lead to better outcomes. Another important direction of future inquiry will be the characterization of relationship between the urinary microbiome and other commensal bacteria, like a crosstalk between the gut microbiome and the GU microbiome, so on and so forth. It is also not known if the microbial domain is different between the upper urinary tract versus lower urinary tract, as well as that in the prostate. Coming to the microbiome and the prostate cancer, we have to understand that the prostate microbiome is very dynamic based on the individual as well as the interventions that the doctors provide. Depending on their sexual preferences, sexual habits, their hygiene levels, the microbiome of the prostate has been shown to be constantly changing, and this can also be impacted whenever we do a urological intervention like a cystoscope or even a transrectal guided ultrasound biopsy to document prostate cancer. Now it has been, it is still in press, but this is one of the nice articles that I found out that this will be the largest, documentation of the prostate microbiome using a metatranscriptomic approach based on the total RNA sequencing data, which is getting published, looking at 94 patients who underwent radical prostatectomy. What was identified in this paper was that there was considerable dysbiosis and certain bacterial species like the Bacteroides fragilis, and I really can't pronounce this, beta herpes virus, Staphylococcus saprophyticus, had a, whenever this was there in normal abundance, then the risk of the prostate cancer was low. Whenever these bacteria and viruses were low, the aggressiveness of the prostate cancer was high. There are several papers who have looked at the microbiome of the prostate cancer. Yes, I'm completing, sir.
Shivat, one is, I think your audio is, yeah.
Am I audible?
Yeah, Shivat, your audio is a bit patchy, and I think we need to wind up also.
Yeah, I'll wind up in a couple of slides.
Okay.
So I just wanted to highlight that, now of late, identifying this microbiome has been made easier by using the NGS or the next-gen sequencing platform. And we can now not only identify just the bacteria, it can also, we can also identify the viruses, fungi, and other parasites very easily using the NGS platform. I'll just skip the kidney. The one point that I wanted to make with the microbiome and kidney is the impact that the microbiome is going to have whenever we put patients on PD-1 therapy. So again, this is a very interesting cartoon. For lack of time, I am just avoiding this. This is one thing that I really wanted to highlight, how the gut microbiome impacts treatments. A lot of chemotherapeutic agents. From the urological perspective, what has been studied is gemcitabine, etoposide, and cisplatin. All of these drugs can be aggressively metabolized with gut bacterial translocation. It's not that we have given these drugs orally. Whatever chemotherapy is in the capillary gets into the gastrointestinal tract, and because of induction of dysbiosis, the chemotherapeutic efficacy is reduced. Not only that, there can be an outgrowth of pathogenic microbes which can propel carcinogenesis. Not only will that metabolization and inactivation of chemotherapy happen, there are certain bacteria which can also reactivate chemotherapy or the metabolite of the chemotherapy to enhance drug toxicity. So the impact of microbiome on malignancy as well as therapeutics is probably very huge and still as yet underexplored. I think with passage of time, microbiome and their understanding will impact GU cancer treatment considerably. Thank you very much.
Thank you, Shivat. So that was really informative. And I think we came from the stratosphere to the earth, and then we saw the most populous creatures on this earth. Shivat, so this normally when we talk about organisms, we would generally think of cultures actually, culturing them and then identifying them. So does that work over here? And if not, then what is the best way to identify them? And because they're going to be such a vast array of these microbiomes.
Yes, thank you, Gagan. So basically, all these bacteria are identifiable, but historically, the microbiologists and the clinicians have broadly identified a set of microorganisms which often trouble the patients, and the culture media that makes them help grow will by default are designed to suppress the growth of these saprophytes. So it is if you start opening up the culture media and try to make it more and more neutral, we can identify a lot of bacteria growing in even apparently sterile urines. But that is going to be really complicated, tough, time consuming, because you will find a plethora of microbes when you just try to keep the media very neutral. So the idea is to use next-gen sequencing. It is expensive at the moment, not freely available, but because we know the genetic makeup of these microbes, using next-gen sequencing will be the easiest and the best way to identify this flora.
So somebody with an NGS system in place would be able to would we get the okay. Thank you, Wilson.
Okay, thank you. Thank you very much, Dr. Srivatsa and Gagan. Thank you so much. We really had, I expected these to be a basic talks, and they really were good basic talks for us. I see that we were approximately 50-60 people, but looks like these were interested in listening to the basics in these particular omics. And as we see, we began with transcriptomics, proteomics, and metabolomics, and it looks that in future we are definitely going to get an advantage with respect to finding out what is the diagnosis, what is aggressive, what is non-aggressive, how we can maybe monitor, predict, prognosticate, and a lot of things in that. Then radiomics, I am sure, will also chip in and tell us about again how to do the same things again with the help of radiological data and put in. And the last talk by Dr. Srivatsa, where microbial flora, I mean, I was also listening carefully to that. It's like we have so much microbial material in a human body, and particularly if it affects our daily systems, right from the etiologenesis of cancers to probably enhancing or improvising treatments in cancer with respect to the microbial flora, with respect to various cancers, as Gagan was mentioning, into with analysis. I guess we would definitely have a future full of all these research and translational activities going on to help us reach patients with these kind of diseases. Any comment from anybody is welcome, because after this I am extremely sorry for the 15-minute delay, because I strictly wanted it from 7:15 to 8:30, but we are still finishing in a very good time. And I thank everyone who is attending this. I hope they are carrying something away, which was the intent of planning this meeting. Everybody is carrying something away from this. Thank you. Any comment is welcome here from anyone. You can just unmute, and I request people to unmute everybody.
I think Gagan, I hope I'm heard.
Yes, you are.
Yeah. A wonderful session we had. A small announcement I have to make. I'm convening the next session, which will be on radiology in uro-oncology. The topics have been decided and discussed. And once we have permission from the USI and a date, next month we will be doing on imaging in uro-oncology. So that's the information from me. Thank you, everybody.
That would be our next talk. Dr. Mallikarjun wants to speak.
Thank you, Ganesh. It's a wonderful thing. I actually came down and then glued myself on this. I know that these are difficult topics, and people who spoke have been neck deep into their work. We know when they speak, it's not that they actually brought out some pieces from here and there and then patched it up. So it was a great thing. And I think one thing which I realize about all this is there is a huge area in where we need to look how we all actually look at cancers. I know we all would like to operate, but I think we also would like to see our patients come back and say, I am happy, thank you very much. So I think these are the ones which help us in the patients telling us, okay, thank you very much, sir, that you have done a great job on me. And probably we also look at predictions for the next generation of the cancer patients. So anyway, this was a great initiative. I think forget about how many people are there, but people are there from the first start. I don't think timing is an issue. Doesn't matter. Half an hour we are here for this talk. We are not for this topic. We are not here for the time. Thank you.
Absolutely.
All the
Thank you, sir.
segment
And I
two.
again thank all the speakers, because it takes a lot of efforts to create and present these kind of talks, and you all were really nice. Thanks.
Moderators were also perfect. They actually had also done their homework. That was the great thing here. So it's not that the moderators came and then said, okay, you say this, you do that. But they all had done their homework, and it was great to have them here. Thank you all.
Thank you. So we end the meeting. Thank you, everyone, everybody. Good night. Thank you.
Santosh, yes, the next talk would also be interesting. We'll be there for that.
Inshallah. Fine.
Come on.
Okay, good night. Bye.